In the previous session, we have learned about PBR322. PBR322 is a classical vector. We have used it for many applications. But right now, the most widely used plasmid cloning vector might be the pUC category of vectors. pUC category of vectors is actually a upgraded form of PBR322. Several features and parameters of the PBR322 is modified to create pUC18. So the most prominent vector in pUC plasmid is actually pUC18, and main advantage of this pUC18 is like it need only single step of screening. In the PBR322 we need to do plate the transformed cells in one plate and then do a replica plate. Then we have to compare both plates to know that which one is recombinant. It's a two-step process. But in pUC class of vectors we need only a single step for isolation of this recombinants. It was developed by Joachim Messing from the University of California in 1982. So the P in the pUC stand for plasmid. UC is actually the University of California. So that's where the plasmid is developed. Another advantage of this pUC plasmid is like it is a high copy number plasmid. The origin of replication is modified so that the copy number is about 500 to 600 copies per cell without chloramphenicol amplification. In case of PBR322, we need a chloramphenicol amplification to increase the number of plasmid. But in the pUC class of vectors, we don't need such type of amplification. And most common type of pUC plasmid vector is pUC18. So here you can see the pUC18. The pUC18 has a size of 2686 kilobase pair, approximately 2.7 kilobase pairs. So if you think about the carrying capacity, it is size is less, so it can carry more DNA. So as the size is 2.7, it can carry about 7.3 kilobase pairs of DNA. So the carrying capacity of pUC18 is more than PBR322, just like an upgrade. And if you look at the gene, it has ORI, but it do not have ROP3 gene. So the reduction of size is one reason for reduced size is the ORI. Then it has the ampicillin resistant gene, same as PBR322. And instead of tetracycline resistant gene, it has something else. It has a LAC-C gene, LAC-C dash actually. So it's have a LAC-C dash gene. This LAC-C dash is the gene in the lacopron. You might have studied that. So the lacopron is having three different genes, X, Y, and Z. So here we have a modified form of Z, which is called LAC-C dash. It's actually code for beta galactosidase, but this LAC-C dash is code for a small portion of this beta galactosidase. So instead of tetracycline resistant gene, we have LAC-C dash gene, and we have something else in this site, that gene, that is multiple cloning site. So in the PBR322, you have seen that the resistant endonuclease sites are spread across the plasmid. So we can see the resistant endonuclease sites everywhere. But here you cannot see that. All the resistant endonuclease recognition sites are packed into a small region which is called MCS, or multiple cloning site. It's also called polylinker DNA. So here you can see it has the resistant endonuclease site for HIN3, SPH1, PSG1, then HIN2, XBA1, BAMH1, SMA1, KPN1, SAC1, and ECOR1. So all these resistant endonuclease recognition sites are packed into a very, very small region which is called multiple cloning site, which is inside the LAC-C dash gene. Okay. So here you can see in more details. So the same figure in different color, the ampicillin resistant gene, origin of replication, then LAC-C gene. Inside the LAC-C gene, we have the multiple cloning site, and this is a magnified representation of the LAC-C dash gene and the multiple cloning site. So this one is multiple cloning site. All the resistant nucleus recognition sites are packed into this particular region. Actually, they have another plasmid called the pUC19. So in the pUC19, the orientation of multiple cloning site is reversed. So here we can see the echo in the beginning, but in the pUC19, the echo is here. So the 5' to 3' directional change is there. So that's only difference between pUC18 and the pUC19. Other than that, everything is the same. So the multiple cloning site is one of the main advantage of this PBR322 pUC18 vectors when compared to PBR322, because the cloning sites are packed into a small region. This Polylinker DNA or multiple cloning site is a synthetic piece of DNA with a large number of resistant nucleus recognition site. One other feature of this multiple cloning site is that the multiple cloning site itself do not disturb LAC-C gene. So the LAC-C gene function normally with the multiple cloning site. But if you are inserting anything extra into the multiple cloning site, it will disturb the expression of LAC-C-. So if we have a normal PBR322, the multiple cloning site is not affecting the LAC-C function, so this LAC-C will be produced. But if you insert anything, if you are inserting a foreign DNA into the multiple cloning site, it will disturb the LAC-C. That's how we place the multiple cloning site. Okay, so inserting a foreign DNA fragment affect the LAC-C gene. LAC-C gene is actually producing alpha peptide of beta galactosidase. So that's one advantage. Another advantage is the single step screening. Single step screening of PBR322 is possible by the blue white screening method. Okay, so in the PBR322 we have ampicillin resistant gene. So for the selection we will use ampicillin resistance and the blue white screening. The blue white screening is possible by the beta galactosidase gene. Okay, so beta galactosidase is actually encoded by LAC-C gene. Okay, beta galactosidase protein is encoded in the LAC-C gene. So I would like to, this beta galactosidase is a huge protein with the size of 468 kilodalton. That's a huge gene. So we cannot actually insert that big gene into the plasmid. So we have done a small trick. So I will explain that. So the LAC-C gene, as I said, it's a big gene. Some of them is having a mutation. Some bacteria has shown a mutation. So this mutation, with this mutation, what happened is like it do not form a functional protein. So the LAC-C gene is actually a tetramer. So four subunits combine to form the functional protein. But with this mutation, if it is mutated in some areas, it will not produce the functional protein. So such a mutation is called the omega peptide. Okay, so the omega peptide is a mutated form of LAC-C gene. It's a large peptide, but it is not active. It is inactive. It is not functional. And we have found another mutation. The another mutation is called the LAC-C dash. LAC-C dash is the one in the pUC18 vectors. So LAC-C dash is actually producing a very small peptide, only with the 50 amino acids. So it's called alpha peptide. The thing is that when omega peptide, which is large, and the alpha peptide, which is small, when they combine together, when the alpha and the omega combine together, it will make a functional protein. So that's we call the alpha complementation. So the original protein, the LAC-C protein, mutated protein is omega peptide, which is large, and the alpha peptide, which is small, only 50 amino acids. When the alpha and omega combine together, they make a functional tetramer, and they will make a functional beta galactosidase, and this process is called the alpha complementation. So the alpha peptide can actually complement the functionality of the omega peptide to make a functional beta galactosidase gene. So what we are doing in the pUC18 class of vectors, the host cell, the host bacteria in their genome, they will have the omega peptide. They will make the omega peptide, but the omega peptide is inactive because of the mutation. And the pUC18 vector is having the LAC-C gene, LAC-C dash gene. The LAC-C dash gene is actually producing the alpha peptide, which is small. So as it is a small protein, the small gene is needed for this encoding this gene. So our pUC18 is having LAC-C dash gene. LAC-C dash gene produce alpha peptide. The alpha peptide produced by the plasmid will combine with the omega peptide produced by the bacteria and make the functional protein. So that's how it's work. So the whole process is called alpha complementation, and we can say it's a intermolecular complementation because here is another representation here. So this one, the bigger one, is the inactive omega peptide produced by the bacteria, and this one, the small one, is the alpha peptide produced by the pUC18 vector. So in the pUC18, this gene, the LAC-C dash gene, is actually producing the small peptide, and the bacteria is producing the large one. So when they combine together, what happens? It will provoke active beta galactosidase. The large one itself is inactive and the small one itself is inactive, but when they combine together, they will get a functional protein. So if you want to see the real image, here how it look like. So this is the omega peptide which is inactive. So when it combined with this small peptide which is alpha peptide produced by lac gene, it will form a tetramer, and this tetramer is actually able to convert the lactose into other compounds. Okay, so it is actually working. The beta galactosidase will work in the presence of alpha complementation. So what we will do for the cloning, the PBR322 pUC18 class of vectors, all clonings are done in multiple cloning site. As I said before, multiple cloning site do not disturb the LAC gene. So an undisturbed plasmid will produce functional alpha peptide, and this functional alpha peptide will combine with omega peptide and produce active beta galactosidase. But if you insert something into the LAC gene, what happens? The alpha peptide lac gene loses its activity, and the alpha peptide will actually uh, like will not produce. Okay, the production of alpha peptide do not happens, so there won't be any alpha complementation. The cell do not produce functional beta galactosidase gene. So if a bacteria is having the complete unaffected, or maybe the non-recombinant plasmid, it will produce alpha peptide and it will produce functional beta galactosidase gene. But if the bacteria is having a transformed recombinant plasmid, it will have a extra foreign DNA in the multiple cloning site, which will disturb the alpha peptide production and which will not produce a functional beta galactosidase gene. So depending on whether it is producing beta galactosidase or not, we can say whether it is recombinant or not. So in the case of alpha complementation or blue-white screening, in the case of for beta galactosidase, the inducer is as you know, the for the lactoperon inducer is lactose or allolactose. So that's not a good inducer. So we are using some other type of inducer in the case of experiments. So we are using IPTG. IPTG is the inducer for the, is a chemical which is used as an inducer for the lactoperon and for the beta galactosidase gene, and the substrate we are using is called the xgal. Okay, the xgal, the full form of the xgal is actually 5-bromo 4-chloro 3-indolyl beta D galactopyranoside. So that's abbreviated as BCIG or simply the xgal. So xgal is the substrate for beta galactosidase. The peculiarity of the xgal is that the beta galactosidase will cleave the xgal into a colored compound. The xgal is a colorless compound. So once it broken with the help of beta galactosidase, it will produce a blue colored compound. So colonies, the cells which is producing beta galactosidase gene will appear in blue color, and which is do not produce the beta galactosidase gene will be in colorless. Okay, so this is how it's happened. So xgal is cleaved by beta galactosidase, the functional beta galactosidase. First it is converted into 5-bromo 3-chloro high hydroxy indolide, then it is non-enzymatically it is converted into a colored compound. The second one is blue colored compound, which is called a 5,5-dibromo 4,4-dichloro indigo. So that's how the cell become blue in color. So the cells which is if our medium is having xgal and IPTG, the cells with the beta which are producing beta galactosidase gene will be blue in color. Okay, so if it is not producing beta galactosidase, it will be transparent or white color. Here is a representation. So this is the genomic DNA. The genomic DNA is actually producing omega peptide. Our plasmid DNA is producing alpha peptide, and they combine together to form the functional beta galactosidase gene. Once it is functional, what it do? It will cleave the beta xgal to produce the blue color compound. So come to the experiments. So we have different possibilities here. Uh, so this is our pUC18. We have the foreign DNA. So when we try to insert the foreign DNA into the plasmid, we have different possibilities. Okay, so maybe the foreign DNA do not accept the DNA. Okay, if the cell do not get cloned in the multiple cloning site, what happens is like the multiple cloning site will be intact. If the multiple cloning site is intact, it will produce alpha peptide, and alpha peptide combined with the omega peptide to produce functional beta galactosidase, and the cells will be blue in color. Okay, so if the insert is outside multiple cloning site, some cases, so here we want to insert in the multiple cloning site, but if it is inserted outside, still they will produce functional beta galactosidase and they will be blue in color. But if the cloning was successful and we are able to insert our foreign DNA into the lac-C dash gene, it will disturb the multiple cloning site, and the disturbance of multiple cloning site do not produce alpha peptide, and so there is no complementation. Those cells will be white in color. So uh, the bottom line is like, in the case of pUC18, if the cell has got the recombinant plasmid, it will produce white colonies. Okay, if it is getting non-recombinant plasmid, it will be blue in color. So we want the recombinant plasmids. So recombinant plasmid cells will be white in color. Okay. Here you can see the scenario. This is our PBR322, and this PBR322 is cloned. When we are cloning, this foreign DNA is inserted into the polylinked DNA. And there are three different possibilities, just like in the PBR322. Either the cell do not take up the DNA, the non-transformed cells, or it can take up the recirculated plasmid without the DNA insert, or it will have the foreign DNA inserted plasmid or the recombinant plasmid. So how do we distinguish between these cells? The first one, the non-transformed cells. The non-transformed cells will be sensitive to ampicillin, because the normal Escherichia coli is actually sensitive to ampicillin, but if the cell is not transformed with the pUC18, it will remain sensitive. So the untransformed cells will be sensitive to ampicillin. Our medium is having ampicillin. The selection medium we are using will have ampicillin, XGAL and IPTG. So as the medium is having ampicillin, the non-transformed cell do not able to grow. So that one will be eliminated. And second option is the cell which is transformed with the plasmid, the non-recombinant plasmid. The plasmid is having all genes but do not have the foreign DNA. So with this plasmid, they will be able to resist the ampicillin. The plasmid is having ampicillin resistant gene, but it do not have the foreign DNA. As it do not have the foreign DNA in the multiple cloning site, the lac-C dash gene will be active. It will produce alpha peptide. This alpha peptide will combine with the omega peptide produced by the bacteria and produce a functional beta galactosidase gene. As our medium is having IPTG, it will induce the expression of beta galactosidase gene, and it has the XGAL. The XGAL is cleaved by the beta galactosidase gene produced by alpha complementation, and this colorless XGAL will be converted into blue colored compound, and this blue colored compound will be accumulated inside the cell. So the cells will be blue in color. If the bacteria is transformed with unaffected or non-recombinant plasmid, the cell will turn blue. And the final one, the one we want, the plasmid with the recombinant DNA. The recombinant DNA is cloned into the multiple clonal site. As we clone the DNA into the multiple clonal site, the multiple clonal site will disturb the beta galactosidase gene or lac-C dash gene. So it do not produce alpha peptide, so the complementation do not happens. There won't be any functional beta galactosidase gene, so it will be white in color. So the bottom line is that the cells we are seeing in a plate which is containing XGAL, IPTG and the ampicillin, this plate, the cells which is growing in blue and white color. The blue colored colonies having plasmid which do not have the foreign DNA, and the white colored colonies is actually having the plasmid with the recombinant DNA. So that's how we identify the pUC18 transformed cells. And here you can see the plate. So here you can see the blue colored colonies. The blue colored cells are actually the one we do not need, because it has having the recirculated plasmid and unaffected multiple clonal site. The white one is having recombinant DNA. So all of them is having ampicillin resistant gene, that's why they are able to grow. So we will take up these cells and we will culture it in the separate flask to get the colonies. And advantages of pUC18. pUC18 is, as I said before, it's an upgraded form of PBR322. So it has some advantages over PBR322. The first one is the DNA fragment can be cloned into a variety of restriction endonuclease site, because it has the multiple cloning site. The cloning will happen only in that particular site. Second thing, recombinant screening is easy. We need to use only one plate, the one plate with XGAL, IPTG and ampicillin. The cloning will be done in a day. We don't have to do the replica plating. That's an advantage. And finally, instead of tetracycline resistant gene, which is a huge gene, we are using a alpha peptide. Alpha peptide is very small, So the overall size of the plasmid is low. So the carrying capacity is high when compared with the PBr322. One factor we need to consider, for the PBr322 we can use any type of Escherichia coli for the experiment, but pUC18 we cannot use any type of Escherichia coli. The normal Escherichia coli is actually producing the beta galactosidase gene. They have the lactoperon, so that cannot be used for pUC18. For pUC18 we are using a special strain of Escherichia coli which is called the DH5 alpha. DH5 alpha is actually developed by Douglas Hanan to maximize the transformation and aid alpha complementation of the pUC18 vectors. So these cells are competent cells. They are we can actually transform the cells using calcium chloride method. We'll be learning the calcium chloride transformation in the next chapter. So the advantages of the features of the DH5 alpha is having, they have three different mutations. So these cells are actually made for higher number of recombinants. So they have mutations. The first mutation is END-A1 mutation. It's with this mutation the intracellular endonucleases are deactivated. So it do not cleave the upcoming DNA. Second mutation is HSDR17 mutation, which eliminate the endonucleases, including ECHO-K1 precision modification system. So our DNA, the plasmid DNA, do not cleave inside the cell. Finally, it has a REC-A eliminations. It prevent the homologous recombination. So all three mutations is actually improving the recombination frequency, because a normal Escherichia coli will have the endonucleases which is designated to cleave the foreign DNA. But the DH5 alpha do not have that one. We have eliminated those genes so that the transformation frequency will be high. On top of that, we have lambda lac M15 gene. This is actually code for the omega peptide. So alpha complementation is possible in the DH5 alpha. So taken together, pUC18 or pUC class of vectors is a upgraded form of PBr322. When we are comparing with the PBr322, the size is less, and more importantly, the screening process is a single step process. We are using the blue-white screening, and but the disadvantage is that we cannot use it for any type of Escherichia coli. We need specialized form of Escherichia coli called the DH5 alpha for the cloning of pUC18, and it is widely used or commonly used in laboratories now. So that's all for pUC18. Thank you very much for watching. Thank you.